Hello, and thanks for joining us today for this webinar, the second in a two-part series on oligonucleotide discovery and development, brought to you by Pharma Intelligence and sponsored by WuXi AppTech. Today's session is focused on oligonucleotide drug development, CMC, and will have presentations from experts involved in the manufacture and formulation of oligonucleotide therapeutics. My name is Mari Gerard, Managing Editor for Custom Content at Pharma Intelligence, and I will be your host today. Before we jump into the presentation, let's go over some quick housekeeping items. First, if at any time you're having technical difficulties, click on the Test Your Connection button, and this will take you to a connection checker and FAQs page. If you are experiencing any technical issues, there is a chat now function in order to go through to the support team who can assist you. Want to enlarge the presentation? Just click on the Maximize icon or drag the lower right-hand corner of the presentation window to enlarge. There will be the opportunity for Q&A at the end of the presentations, so if you have questions, we want to hear from you. To ask something to our presenters, simply type your question into the question window on the bottom of your screen and then hit Submit. We'll be answering as many questions as possible during the Q&A, and if there's extras, we'll try to answer you after the webcast via email. Because we know you'll want to listen to this webinar again, today's session is being recorded and will be available to view on demand in around 48 hours. I would now like to introduce our speakers for today's session. You can see their full bios by scrolling down below on your screen. First, we'll have Mark Edbrook, an independent consultant. Then we will have William Fang, Vice President at WuXi STA, a WuXi AppTech company. Then we will have Lu Chan, Director for CMC Project Management, also at WuXi STA, a WuXi AppTech company. And finally, we will have Mike Webb, an independent consultant. And with that, I will hand things over. Mark, the floor is yours.
Hello and welcome everyone to this webinar about oligonucleotide drug development. And welcome back to those of you who attended the first webinar on oligonucleotide drug discovery. My name is Mark Edbrook, and I've been working in the field of oligonucleotide therapeutics for over 25 years. So I have a question for you, and that is, what is exciting about the drugs depicted on this slide? And the key is that these are all oligonucleotide-based medicines that have been approved in the last 2 years. And this demonstrates the extraordinary progress that's been made by this modality of drug in recent times. In fact, what it now means is that there are more than 16 oligonucleotide therapeutics that have been approved, and that is not including N-of-1 drugs like Millicen. And these drugs include a range of different types of oligonucleotide drugs, so includes single-stranded and double-stranded oligonucleotides, different routes of delivery and different formulations. And as listed here, oligonucleotides can be used in many different ways in order to affect gene expression. So for example, they can be used as gapmers to lead to mRNA degradation. They can be used as aptamers to bind to intracellular and extracellular receptors. They can be involved in nuclear steric blockage for splice skipping or splice switching. They can be used to block the assembly of RNA binding factors. They can be involved in toll-like receptor activation in innate immunity. They can act as antagonists or mimics of microRNAs. They can be used as agamers to block translational initiation. And of course, there is RNA interference using the RNA-induced silencing complex. And so oligos can be used as small interfering RNAs or short hairpin RNAs. And I do apologize, I have gone the wrong way. And that is demonstrated in this elegant diagram from a review paper by Susan Hammond in EMBO Molecular Medicine last year. And so going from left to right, it shows the mechanism of action of gapmers leading to RNA degradation, aptamers, antisense oligonucleotides, TLR binders, microRNAs, agamers, and siRNAs and short hairpin RNAs. And if you want to learn more about how the oligos work in different modalities, then I would recommend that you look at this review. Similarly, as you will hear in the following presentations and also in the previous webinar, which I also recommend that you attend, the key to this progress has been the evolution of oligonucleotide chemistry and also the increasing in biological understanding of how oligos work. And again, this is summarized nicely in this diagram and also in this review by Anastasia Korova and John Watts in Nature in 2017. And again, I would recommend looking at this article if you want to see more about how oligonucleotides work. So these webinars are going to cover key aspects of therapeutic oligonucleotides in this two-part webinar series, and it'll cover oligonucleotide chemistry and design, biology, DMPK, toxicological assessment, and in particular in today's webinar, manufacturing, purification, and formulation. What I would say is please enjoy the presentations and be ready with any questions which you can put into the Q&A chat, Q&A box at the bottom of the screen. Thank you very much.
Thanks everyone for joining this webinar. And some of you might have listened to talks by our discovery focus yesterday. In my today's talk, I will mainly focus on the introduction of this oligonucleotide CMC platform. A very quick glance of the basic structure of nucleotides. In terms of the drug design, chemical modification is one of the most common and effective approaches to improve oligonucleotide drug delivery. Modification can be on the nucleic acid backbone, the base, or the ribose sugar itself, such as 2' hydroxy substitutions, constrained sugaring, or even replacement of 5' sugaring with 6' morpholino group, which is also called PMO. For the manufacturing of oligo drugs, it employed similar approaches as peptide, which is to utilize the solid phase synthesis strategies to assemble the sequence and use cleavage and deprotation to obtain the crude API and purification or iodization to get a final purified API. For duplex, one additional annealing step is needed in the process. With the current industry demand, WuXi STA has established an end-to-end full CRDMO service platform in this field to cover from discovery, development, to commercial manufacturing at various scales. We have been able to synthesize different types of oligos and related conjugates. Furthermore, we also can work on novel amidites and galvanic material to ensure the safety of supply chain when carrying on large-scale manufacturing. We also established a full CMC platform to be able to work not just API manufacturing, but also drug product, and for support in analytical and regulatory filing. A little later, one of my colleagues in formulation group will talk about the LMP technology we have worked on. This map shows the location of our each group. Tianjin side is where our oligo discovery group of folks are located. Changzhou is mainly for process development and larger scale manufacturing. Shanghai for pre-formulation and formulation development, and WuXi is for fill finish larger scale production. Now let's come back to Changzhou site, which is one of our largest manufacturing campus within WuXi AppTec. Changzhou City is about 1 hour bullet train distance from Shanghai. Our site has an area of over 70 acres, and all our oligo development, manufacturing of API and supporting teams are located here. In top left, there's our first oligo GMP plant, which was commissioned over 2 years ago. This building is equipped with 1 mid-scale OP400 line and a large-scale oligo process line. And in the top right, you can see our second oligo GMP plant, which was just commissioned actually last week. In this new plant, we added 3 more oligo process lines. In regarding our oligo process development, we have equipped significant numbers of OP100 and downstream equipment. Till today, our team has worked on more than 100 oligo projects, covering many types of oligomolecules, utilizing various of modified monomers and building blocks, and with modification at either 3' or 5' end. In terms of manufacturing, we have equipped several sets of oligo lines from synthesis to purification, UFDF, and lyophilization. All the main equipments are Sativa brand. For purification, we also added a reverse phase HPLC unit from Novasap in case some of the projects requiring additional reverse phase purification. For thin film evaporator, we obtained the latest version in the industry with centrifugation capabilities so that we can concentrate final product solution at a much lower temperature and at a higher speed. With the booming interest in the field in recent years, the industry is calling for additional manufacturing capacity. Therefore, we have been dramatically increased our manufacturing capacity. As you can see here, with the addition of a second oligo GMP plant, we now have 3 OP400 lines and 4 oligo process lines. And we have more manufacturing plants scheduled to be ready next year to increase the OP400 line to 4 and oligo process line to 7. For analytical capabilities, we have established extensive capability to handle all oligo API-related testing and releasing service. We will have 9 QTOF high-resolution masks by the end of this year, so that we have sufficient capacity to meet our increased demand from our projects. Currently, we have over 130 analytical chemists to support both our oligo and peptide projects. One of the unique analytical techniques we have been using in the group is to use 2-dimensional UPLC coupled with QTOF high-resolution mask for impurity analysis. With this approach, it can provide detailed impurity profile to our process chemists and dramatically improve efficiency during process optimization. As I mentioned earlier, our team has worked on many types of oligo projects. Here is a typical small interference ionate duplex project we have worked on. The work scope covering from process development, GLP scale-up, and GMP manufacturing. The GalNAc modification is also typical at the 3' end of sense strand. As you can see, the anti-sense strand synthesis is pretty straightforward with good purity and yield at the end, while for the sense strand, due to the stability issues of specialized linker and some steric hindrance of GalNAc group, it did require some additional process optimization work. But at the end, our team was able to find some suitable process conditions to improve the yield and the final product purity. The data showing here is from a 2.5 kilogram GMP campaign. So till now, we have worked over several dozens of small interference RNA duplex projects with typical GMP batch size ranging from a few hundred grams to a few kilogram scale. And another type of oligo molecule is called PMO. Due to the high cost of PMO monomers, we developed our higher loading resin linkers and to use less equivalency of monomers while maintaining high purity crude product. This has dramatically helped to reduce the manufacturing cost for this type of molecule. We developed several types of linkers at the 5' end to provide client with more options to further for further 5' modification. In addition, we used the monomers with alternative protection groups and developed comparable cleavage and deprotection process to circumvent the IP issue in this area. so that we can work with anybody in the industry without worry of IP infringement. Till now, we have established a 6 GMP line at the largest 100-liter scale. Now we are adding additional line up to 200 liters. This slide shows a case of a conjugation project with peptide to PMO. As you know, we have both peptide and oligo CMC team. So we can mobilize our peptide, oligo, and analytical team to work together in parallel to reach a good efficiency. We have been working with most of the industry-leading research company in this field and have successfully completed over 35 integrated PPMO projects so far. Another case showing here is our unique integrated amidite, GalNAc, and oligo development and manufacturing project. Again, we have internal dedicated amidite and GalNAc team to work together with our oligo team for easier material inter-group delivery, for troubleshooting, and for method transfer. In here, the 2-fluoroamidite is commercially available, but it is very expensive and long lead time at a larger quantity. Producing it in-house is to increase project flexibility and to reduce risks of unexpected supply chain interruptions. Above is all I have to discuss today, and thanks again for your time to listen. We will have a separate Q&A section a little later, and we can answer any question you may have by then. Thank you.
Hello, everyone. Welcome to the webinar. Today, I'm going to talk about the formulation, drug delivery aspect of oligo and the nucleic acid therapeutics. Also going to introduce the current LMP platform at WuXi FDA. We're looking forward to helping our current and future customer on the oligo therapeutics development and bring more breakthrough medicine to the patient. So at first, let's take a look at the mechanism of the nucleic acid therapeutics. Unlike the traditional small molecule drug, which deal with the protein function to treat disease, the nucleic acid drug RNA and DNA target the upstream gene expression, such as the transcription and translation. A lot of people believe this therapeutic modality will bring a great platform technology to treat disease and even for traditional undruggable therapeutic target. So as I will discuss in the later slide, if we can solve the delivery challenge, one can simply change the sequence of the oligo without a significant change in the physical chemical property of the drug molecule itself, and be able to treat a large variety of disease. So here is a list, several nucleic acid-based drugs and their mechanisms. For example, antisense at a single-strand short DNA molecule, they bind to the mRNA and inhibit its translation process. siRNA form RISC complex in the cell and cut mRNA sequence specifically and inhibit the protein translation. So more recently, significant development of mRNA vaccine to target the ribosome to translate antigenic protein and trigger the immune response for the virus. So as aforementioned, oligo drugs provide a great advantage over traditional small molecule drugs. The formulation delivery challenge are tremendous. There are still a rate limiting factor to create an effective therapy, and due to the complexity of the formulation, a single solution is unlikely. So here are some of the hurdles for deliver oligo drug in vivo. So first, the delivery system has to encapsulate the oligo efficiently and protect the drug while in the transit. Secondly, they have to get into the target tissue through the circulation or other route, and the targeting to the specific cell type as designed. And one of the critical steps is to release the payload to the action site once in the cell. For example, the cytoplasm for the mRNA target. Over the decade, people have been developing all types of delivery systems to overcome the challenge. For non-viral delivery system, they can be categorized into these 4 types of system. First, the conjugation, to conjugate oligo with the targeting or other functional molecules, such as the cholesterol, peptide, sugar molecule, such as the GalNAc. Second, lipid-based system, such as the lipid nanoparticle, LMP. This involves development of functional lipid, formulation optimization, and also the manufacturing process control is very critical. The third is the polymer-based system, which has a greater flexibility for structural design in chemistry to customize delivery need. There are also the so-called hybrid system, which involve the polymer lipid to give a more flexible formulation design. So today we will focus on the LMP system, talk a little bit about the key development in the field, and introduce our LMP platform at WuXi FTA. So for lipid nanoparticle or LMP, there are huge success on mRNA vaccine development in the last couple of years. More and more companies are investing in this technology and seeking the breakthrough medicine beyond the vaccines. As many of you already know, that LMP is not new in the drug product development field. The liposome drug has been around for many decades. The traditional liposome delivery concept is shown on the left-hand side. As you can see, it's a typical lipid bilayer vesicle, which includes the aqueous phase inside. The hydrophilic drug can be encapsulated inside the vesicle, while the hydrophobic drug can be loaded into the hydrophobic lipid bilayer. For nucleic acid-based oligo RNA, due to the negative charge of phosphate of the molecule, cationic lipids are used to encapsulate the oligo through the tartar interaction, and the complex will further wrapped around with structural lipid and PEG lipid. So the microstructure is more like a solid core shell material with the oligo domain inside the particle. So people normally call this type of formulation LMP instead of liposome. To develop such a complex multi-component system, there are two key areas trending in the field. One is to develop innovative functional excipient for better therapeutic index, like high efficacy and low toxicity. The other aspect of the effort is to develop the efficient process to assemble such multi-component system. So they are new material and a new process that drive the innovation. Regarding the new material in LMP system, a lot of effort were put on to develop the novel lipid. This slide shows that the concept of ionizable lipid, which are the key functional excipient in the recent developed LMP system. Ionizable lipid are new generation of cationic lipid with specific pK. At the neutral pH, there is no charge, while at the lower pH, the tertiary amine in the molecule become positive charge. This can help encapsulate the oligo at the lower pH through the tartar interaction, while at the neutral pH without positive charge, and they can lower the toxicity of the system. Also, the lower pH inside the endosome, after entering the targeting cell, they can convert to the hexanyl H2 phase and cause the membrane fusion and disruption. So this will facilitate the endosomal escape to deliver the cargo into the cytoplasm. Here are three significant examples for ionizable lipid. So first one is the MC3 lipid for Onpattro, and the ALC0314 for Pfizer-BioNTech mRNA LMP, and the SM102 lipid for Moderna mRNA LMP vaccine. So unlike traditional thin film hydration coupled with the side extrusion process for liposome, current LMP assembly apply the principle of ethanol mixing method. As shown in this diagram, all the hydrophobic lipids are dissolved in the ethanol, and the oligo are dissolved into aqueous buffers. The organic ethanol solution then mixed with the aqueous phase at first. The cationic lipid captures the negative charge oligo, then other structural lipids wrap around the complex to form the nanoparticle. The particle size and the encapsulation efficiency can be optimized by the design of the mixing device and the process to control ethanol-water mixing ratio and the organic phase dilution rate. One of the keys to control the process reproducibility. We also can apply the DOE approach to optimize the process. According to the FDA and the EMA, here's a critical quality attribute for the LMP product. In particular, the testing items such as the lipid content, encapsulation efficiency, particle size, and in vitro release. So also back in 2018, FDA published the final guidance for liposome drug product, which is a great reference for CMC aspect of LMP drug product development. In the following slide, I will go over the LMP platform development and the capacity at FTA. Hope to provide detailed information on our platform and look forward to future collaboration with the current and the new customer for oligo LMP drug product development. So first, based on the ethanol mixing method, we are using a multi-channel macro mixing device to produce LMP assembly with the robust scalability and the reproducibility. With the process optimization and formulation development, it can provide the lipid nanoparticle with various assembly structure. The whole process involves mixing, ultrafiltration, sterile filtration, and fill finish. We're using Onpattro siRNA LMP as our benchmark drug product for our for our platform development and validation. As a benchmark product manufactured in-house, the siRNA LMP particle has small particle size below 100 nanometer with a narrow PDI less than 0.1, and the purification stereo filtration process has been optimized to support GMP manufacturing. Our GMP system is a modular design system with a high flexibility for the process modification to serve different customer needs. Right now, the setup of the GMP facility has been completed. The whole system will be validated and ready to serve customers by end of the third quarter this year. So this slide shows some typical equipment and instrument for our LMP platform. Here's our multi-channel macro mixing system that includes a mixer and a pump. We also have TFF purification system with different scales. Here's the stereo filtration system. So besides the process optimization on the multi-channel macro mixing step, we also have done a lot of evaluation and optimization work on the TFF and stereo filtrations. In each lab, we also have the Marvin Zetasizer as standard equipment for particle size measurement. In our research lab, we also have different mixing technology for process validations. As I mentioned, we have a fully dedicated analytical team to support our research development and GMP manufacturing of LMP drug product. Here are the list of our testing capability. In addition to the universal testing for injectable drug product, we also have a specific testing capability for nanoparticles, such as the zeta potential, particle size. At the WuXi TA, we also have a fully automated top-line stereo fill finish equipment ready to use for LMP platform. As I mentioned previously, we're taking Onpattro as our benchmark oligo LMP product for our platform development and validation. As many of you already know, the Onpattro developed by Alnylam is the first commercial oligo LMP product. The formulation includes the siRNA target TTR gene expression, 4 lipid components, particularly the MC3 lipid is the ionizable lipid. PEG-DMG is a PEG lipid which will shield the LMP particle with polyethylene glycol layer during the circulation and the deshielding while entering the cell and deliver the payload. So the DSPC and cholesterol as structural lipid in the LMP formulation. As I highlighted here, the siRNA API itself and all the functional lipid MC3 and PEG-DMG were provided by our own SCA team at GMP grade, so which gave us a huge advantage for in-house LMP platform development. Is shown in this slide. So the first step, LMP assembly through the multi-channel mixing, TFL purification step to remove the ethanol, and then do the buffer exchange and the concentration, the stereo filtration and the fill finish. You can see here in the table the IPC result for particle size measurement from a different stage of the process. As mixing stage, during the purification TFF stage, and after stereo filtration. They are very consistent with the particle size average 80 nanometer and narrow PDI below 0.1. Here are the photos of the actual samples at each stage. We also test our siRNA LMP benchmark product according to the specification of Onpattro. Here are the results. So the particle size, zeta potential, encapsulation efficiency, drug loading, organic solvent residue, all the results are consistent with the literature values. We also conduct the stability study for our product. Storage condition for Onpattro is 2 to 8 degrees C. At 3 months, the particle size are very consistent with the initial data. The long-term stability study is still ongoing. To summarize, at the WuXi TA, we are committed to helping our customer to develop novel formulation for new modalities, such as oligo drugs. We have established the technical know-how for LMP and the non-formulation drug development. We have multi-channel mixing technology evaluated and optimized. We also have done a lot of work on TFL purification and the stereo filtration process development and optimization. We have established a scale-up process with a modular design and high flexibility to match individual product need. Our analytical method has been developed for oligo LMP drug product, and our GMP system will be fully validated by end of the third quarter this year. At last, I want to emphasize that our ultimate goal is to provide end-to-end enabling service for our customers. Our LMP platform is a cross-functional platform, and we got a lot of support from our process country team. We have all the key functional lipid and oligo made in-house at GMP level. We also got a lot of support for in vitro and in vivo testing from our other divisions. So a great synergy here at WuXi to help our customer to develop innovative medicine and help the patient in the world. Thank you.
Hello, everyone. My name is Mike Webb. I'd like to thank the organizers for giving me the opportunity of talking to you today. I'm going to focus in only on what I see and in my experience working on the manufacture of oligonucleotides and analysis of oligonucleotides, what I see as the challenges, particularly compared to small molecules. So I will be in this deck focusing on specific slides where we can discuss the challenges that are specific to oligonucleotides. But what I would like to say by way of introduction is with the approval of inclisiran and some of the drugs that pharma companies are working on now, we're seeing oligonucleotides transition from not just being a rare diseases therapy, but being a therapy at a high volume with a very fast-moving supply chain. So as we develop oligonucleotides, we're developing to manufacture for both rare disease supply chains and large therapy supply chains, such as hepatitis and dyslipidemia. So it becomes very important to focus on these challenges because they will become even more acute as we move into this area. So I'll give you an overview of what I see are the challenges for a control strategy. And a control strategy, we'll talk very briefly what that is. I want to talk about some specific analytical challenges for an effective control strategy and what I've seen over the last several years. I touch a little bit on process challenges, and if I have some time left, I would like to talk about some of the experiences working with CDMOs, but particularly in the context of establishing what will eventually be a commercial supply chain as we move from CDMOs supplying clinical material to being highly reliant for a number of companies and patients to be the supply chain. So I think everyone is, hopefully familiar with the manufacturing process, and I'm going to focus specifically on the synthesis and cleavage, less so on the purification, UFDF, annealing and lyophilization. Clearly, oligonucleotides are not as sensitive to the purification and UFDF in terms of their fragility compared to things like monoclonal antibodies. So the challenges tend to be more upstream in the synthesis and of course in the phyloanalysis. This slide really talks about where the analytical challenges come in with respect to oligonucleotides, and in the red I really am talking about the fact that these methods are absolutely critical to the establishment and verification of our control strategy, which is a planned set of controls derived from current product and process understanding that assures process performance and product quality. So we're using analytical methods not just to test in at the end, but to support DOE studies to enable us to establish the optimum manufacturing process and the proven acceptable ranges and the normal operating ranges that our manufacturer will work in, but also to support process development studies. So we need good methods when we start, and we need good methods when we refine our manufacturing process. And of course, we need validated analytical methods and specifications for both our starter materials, our in-process checks, and our final drug substance specifications and methods. And these are critical, and not only they're critical, they are far, far more challenging in a quality control environment. And hitherto, of course, have been involved in quite a specialized analytical environment. So I really want to now just lay out the specific challenges, but then I will focus in on one or two during the rest of this talk. So I think everyone knows that during process characterization, we're dealing with, for example, a 20-mer, it will be something like 80 chemical transformations with no monitoring because it's occurring on a solid state column. until after a cleavage from that column and deprotection. So that is quite a challenge for us to understand process characterization, because we do not know what's going on after each reaction on the column unless we cleave each time off the column, and that's very different from small molecule chemistry. We know that our impurities come in families, so when we talk about an N-1, a failure in the sequence due to chemistry, that potentially could be 19 components for a 20-mer. We know that many impurities are not well resolved, and many impurities collude with the main peak rather than being 3 or 4 impurities that we beautifully separate. We do not have that luxury. We do not have that degree of fidelity in our analytical methods. And we also know that starting materials are critical. Very low-level impurities in starting materials form drug substance impurities. So if you have a G-amidite and you are adding that G-amidite 10 times, a tiny impurity at 0.1%, if it has the same coupling fidelity, will end up contributing a 1% total impurity load within that final drug substance. So the control of impurities at the starting material state has to be very, very exquisite. Potency, specificity of relative assays is very difficult. Ultra pure standards are difficult, and hydroscopic problems, static problems make the samples difficult to weigh, and I'll talk a little bit more about that. Identity testing during characterization is not straightforward. We'll cover that. And we cannot, unless we rule it out as being an issue, we can't naturally make secondary structure irrelevant to our measurement or characterization until we know it is not important. And typically we do not want it to be important because we end up with a bioassay which provides its own problems. This is typically only true for aptamers, where secondary structure is, of course, higher order structure is important to function. But we have to rule it out as something we need to worry about. And normally we're successful, but the regulators expect us to determine that. Finally, we know that some regulatory guidance specifically excludes oligonucleotides. So in terms of impurity qualification, for example, we don't have the luxury of ICH guidance to help us. However, it is not all bad news. We have some advantages. We're using pretty much the same synthetic approach across most therapeutic oligonucleotides, and have done now for a couple of decades or more, which means that we have significant platform knowledge and know-how within chemistry groups, within manufacturing groups, within CDMOs, and most importantly, in the literature. We therefore are using very common starting materials, which means they are well characterized and the process impurities are well characterized. These are not de novo synthetic processes. They're well understood synthetic processes. And we can draw on that platform data to give us a flying start. And then finally, small molecules are very effectively purged because of the way in which we immobilize our growing oligonucleotide on a column, and we wash our reagents from that column. So purification is very good in terms of small molecules. Elemental impurities, solvents, genotoxin assessments are quite common across multiple products. And there are now groups publishing papers that can help us with respect to purging those small impurities. In the absence of ICH, we have some very good publications now on impurities that have been very helpful to new sponsors. And the oligo community has been very, very effective in sharing regulatory experience. And regulators are now seeing multiple submissions. So I really want to dive very quickly through this slide. And we're going to focus mostly on the left-hand side because small molecules, solvents, and genotoxins and mutagens tend to be pretty established and not much of an issue for us. The problems are characterizing impurities from starting materials, process impurities, and degradation products. Firstly, just one quick example of why starting material impurities are so important. And this example here shows you where we can see an impurity, which is basically the protecting group, the DMT group, and the amidite group have been swapped between the 3' and the 5' position. So this will couple, in a sense, upside down, but will give you a totally isobaric impurity, which is likely, using the IPRP methods, to be totally undetectable by the time it gets into the oligonucleotide. Therefore, we have to control that impurity to very low levels in our amidites. So just one example of critical impurities we see in amidites that need to be focused on. I briefly told you that ICH Q3A excludes oligonucleotides, and regulators have been, over the years, working on a case-by-case basis because of guidance, but we now have moved more towards the peptide monograph, and there's an excellent paper by Daniel Capaldi and others in 2017 which set thresholds at 1 and 1.5, which is holding based on specific justifications. So that's really helping us now to move forward. The next example I want to focus on is identity. Typically for small molecules, we have a nice fingerprint in infrared or even NMR, but that fingerprint is just insensitive to sequence and small changes, and NMR spectroscopy is very, very hard to interpret. So we're really in these bottom areas. Retention time is free, but it's insensitive to sequence and structure. Molecular weight is very sensitive to full-length products, but it's insensitive to sequence. So we are then talking about something much more difficult in terms of identity, sequencing by mass spec, but there are other ways of doing it, and we are having a current debate with regulators to try and move to something more QC friendly than high-end MSMS sequencing. So using failure sequence analysis in our crudes, looking at melting temperature, and for an antisense that will be with a complement, or just by showing in a risk assessment that you have such good GMP control in your purification that getting swapping around amidites nucleosides is very, very difficult. So I won't focus on this slide on sequence methods. I move very quickly in the time I've got to just talk very quickly about secondary structure. So we know that although you may have a series of oligonucleotides with the same chemistry, but with different sequencing, different sequences, of course, can anneal and form hairpins, and therefore potentially different structures. Our peptide conjugates or our antibody conjugates in particular will have secondary structure constraints. So you need to think about this. There is good literature and platform data to suggest that antisense and cerners are not secondary structure dependent, but we need to know that it exists. We need to know whether it has any biological significance, whether it be characterized, whether it can be measured, and whether it can be controlled, and in particular, if it is reproducible. I'm going to move very, very quickly now on to another area. Phosphorothioated oligonucleotides are, of course, mixtures of diastereomers. So a 20-mer oligonucleotide will be actually potentially a mixture of up to 500,000 different compounds. And we are seeing from regulators an interest in demonstrating control. And there was very recently a paper suggesting that the failure of Mongersen for Crohn's in phase 3 compared to really good data in phase 2 was because the batches used in phase 3 had a different diastereomeric ratio than the batches in phase 2. I am very skeptical indeed about this paper, and we haven't got time to discuss this, but it is getting attention, and it is important because it is of course impossible to measure such a huge quantity of diastereomers, but it is very, very important that we have some method of showing that our synthesis is reproducible. So if you have limited phosphorothioates, you may see some fine structure in your UPLC data, which will be semi-quantitative, or you can sample and cleave at each coupling at a lab scale and measure the ratio of the two diastereomeric peaks that still have the DMT group on, and repeat this for each addition. And this is a way of showing, particularly if you show over a couple of batches, that we have reasonable reproducibility. And I think there is a debate at the moment that we need to think about. about one, demonstrating this and how far we can go and how important it is, and also what is analytically possible. I'm merely going to show you this separation here, a typical separation where we can see very different to small molecules, where our impurity and purity methods are really challenging. And we can see the small change in chromatography is going to have quite a dramatic difference, particularly on the N+1 and N-1 impurity peaks. And that's going to be really, really critical for us. So it's extremely important to control our methods and to have system suitability controls. This slide really identifies the fact that we are also in the business of measuring by mass spectrometry certain impurities that come under the main peak. I'm going to just finish very quickly on assay, and I really want to talk about very quickly onto this slide. We can do titrations, which are non-specific, we can do UV assays, but for HPLC assays, we have a challenge. Standard is norm for small molecules. The standard is 99% pure, limits to 98 to 102, and we have very good specificity. Precision will give us an RSD of 0.5, which means it's highly accurate and it's great utility. We do not have that for oligonucleotides. We typically have less pure standards. Limits are wider, which is good. We don't have good specificity for the reasons I just said, and we can see that we're probably going to get poorer precision, and therefore our confidence interval is wide, and therefore accuracy is quite inaccurate, and it is really only suitable for checking for large contamination. I would venture to say it is not very suitable for working out where you are with respect to compounding for secondary use. I think the only other thing to say here is that oligonucleotides are also highly hygroscopic. So the water that you have measured may be the water in the sample at the time you did analysis. So it may be important to condition your samples as for any hygroscopic compound, that in the same environment that you're going to weigh it out at the same time as you weigh it out for your assay, you'll need to measure the water so you know how much water is in your sample. I'm going to finish now just by very quickly talking about process challenges. We quite often for rare diseases have limited batch data, and the regulators understand that and are quite happy to include laboratory batches. If you do that, you are going to need good laboratory model for production equipment, which means that understanding the implications of scale up in manufacturing is critically important. And a lot of the, because if we assume plug flow, scale up is potentially more predictable from a chemistry point of view than large volume batch manufacturing, where stirring rates and heat transfer is different. Here, it's really engineering, which is the distribution across a wider column, the packing and lack of channeling. So the distribution of the flow down the column needs to be very similar as you go up in scale. You need good pump performance and you need good monitoring of your D-block process using UV. I think I've pretty much run out of time. All I'd like to say really is that it is really important that CDMOs work as a manufacturing organization would work, providing input into a supply chain, which is a movement from the mindset you would have if you are making clinical supplies. So with that, and given an eye on the time, I'd like to thank you for listening. And I would like to thank all of those pioneering chemists that take on this challenge to pave the way for these exciting medicines. And I think we now have time for Q&A. Thank you very much.
Fantastic. Thank you very much to all of our speakers for those great presentations and insight there. As mentioned, we are going to jump straight into our Q&A, and we have already got a few questions lined up. But just a reminder to the audience, if you do have any questions resulting from those presentations, just enter the question in the question window on the bottom of your screen and then hit submit, and we will answer as many questions as possible in the remaining 10 minutes. And if there's any that we don't get to answer, we will endeavour to do so after the webcast via email. But without further ado, our first question that we have is, in addition to the solid phase, do you have capabilities in solution phase oligosynthesis? And William, it's been directed to you, actually. I don't know if you have any thoughts on this question. You may just need to unmute yourself. Okay, we might have some technical difficulties there with William, and we do have another question actually for Lou. So this question is, what are some key challenges and limitations of LMP, and how do you overcome them?
Thank you for this question. So this question really reflects the current two key areas for LMP development. So as I discussed during the presentation, the first is intrinsic toxicity of cationic lipid. So as it helps the intracellular delivery oligo, but the hydrophobic and the positive charge nature bring the toxicity. So one of the solutions is through chemical modification and design. So for example, the new generation ionizable lipid, which is a less charge at the neutral pH, but the lower pH have a positive charge. And also people apply the degradable acid bond linked to the aliphatic chain with the head group. So the another challenge is the complexity of the manufacturing process for the LMP system. That's why the people are developing all types of mixing device and a mechanism to produce LMP with a consistent quality.
Fantastic, thank you very much. And I do actually have another question, Lou, for you, if that's okay. And that is, yeah. Can you elaborate on your integrated platform of API and DP, and how do the different teams work together?
Yeah, sure. That's a great question. So at FDA, our ultimate goal is provide end-to-end enabling service for our client. So the integration of the API and the DP is very critical for our platform. And there are, I'll say, at least three aspects for this. The first one, the timeline management and the logistics. So because we are under the same system, so we have a huge advantage for the timeline management and accelerate the development for the drug. Another, the second one is the technical support. So for example, as Williams already mentioned in his presentation, let's say analytical method transfer from the drug substance to the drug product is very helpful for the later stage development. The third one is the same QA system, so which is way easier through the process from the beginning, the API to the drug product manufacturing.
Brilliant. Thank you very much for that. I'm not sure if we have William back on the line, but we do have a couple of other questions as well. So one question that has come through is there any issue with attaching GalNAc to the 5-inch ends of oligos, and does this affect the yield? Don't know if anybody would like to take that one, any of our speakers. Might be having some technical difficulties with a couple of us.
Mark.
Oh, thanks, Mark.
Well, I was just going to say that I don't really know, because my understanding is that the attachment of GalNAc is usually on the 3' end, and that's usually done in the solid phase synthesis. So I don't actually know whether or not attaching 5' attachment is quite often done post-synthesis. So I'm not sure whether that affects the yield or not. It may not, but it's as far as I'm aware, it's a different process, putting it onto the 3 different ends, the 3' end, 3 different ends? 2 different ends, sorry, the 3' or the 5'. So I don't actually know if there is a difference, but my understanding is the mechanism of attachment is different.
Fantastic. Thank you very much, Mark. And we do actually have another question addressed to the WuXi side, actually. So Lou, I don't know if this is one you could answer. It says, is it possible for WuXi to generate radio-labeled oligonucleotides, for example, for human microdosing for tissue delivery? Again, might be having some technical difficulties, so apologies, everybody. We have had another question come through, which is actually stating that global capacity in solid phase synthesis is increasing, but the batch size possible is still quite small from a commercial perspective. This means multiple batch analysis. How do you foresee a solution to increase batch size from existing equipment, or does the equipment need to be upscaled in some way? And I'm not sure if any of our panelists would like to take that one. Okay, I think we might have had some technical difficulties. So apologies to our audience for those questions. With that, we'll probably wrap things up. So I would like to thank all of our speakers who have contributed to today's session for their really great insight, and our sponsor WuXi AppTech for making this whole event possible. So on behalf of WuXi AppTech and Pharma Intelligence, we hope that you will have a great remainder of the day, and thank you very much for tuning in today.